you see the screen?
Yes.
Perfect.
Good.
Okay. Okay. So, well, as I was saying, thanks a lot for the invitation, the introduction. Just one thing before I start. So please feel free to interrupt me at any point if there is something unclear. I mean, there are a lot of things I want to say, but I also, if I don't arrive to the end, it's perfectly fine. So don't hesitate to stop me. Just a couple of words from where I come from. So as I said, I work at the ICTP in Trieste, which I guess many of you know, but just this to mention that we have a section, a group working on diverse topics at the interface between biology, life sciences and physics. And we organize many conferences, workshops. So just check the ICTP website and the quantitative life science section website to see what we do, and also visit us. What I want to talk today is about microbial communities. So I thought many of you are familiar with how we study microbial communities nowadays, but it's good to start from the beginning or as a general introduction. And I start from the beginning with this picture of Galileo showing to the Doge of Venice this new invention at the time, for which he was able to see, of course, ships coming to make troubles in Venice, but also you could look at distant celestial bodies. And around the same time where this discovery was made and lenses started to be used, people started to use them to study the very small world that is around us. And this is like one of the first example of a microscope. And people started using this microscope to look at things around. And when they were looking at the like small drops of waters taken from Thames, so very close to you, they started to see these very little monsters. And so the lady was like dropping the cup of tea because of this discovery. Now, this discovery sort of opened the world to these little organisms that lives around us, microbes. And what is incredible about microbes is that they are able to very big adaptation and populate every corner of our planet, including very extreme environments. And as you probably know, they perform essential functions for our planet and our health. What is important for this context is that they live in very structured communities where there are a lot of biochemical reactions which shape their interactions. And well, this is one example. Is actually a simple experiment that you can do at home. If you take like some mud and you add a source of carbon, like shredded papers, and a source of sulfur, like egg yolk, and you mix them and you wait, basically you create this layer of bacteria which performs different reactions. And what you see is a clear gradient which then appears as different colors in this community. And this is it's a different but somewhat similar phenomenon of what happens in ponds, like this one in Yellowstone, where the gradient of temperature in this case creates different layer of different organisms. And so it's very clear we understand very well the structure. And then about 30 years ago, sort of the sequencing revolution came in microbial ecology. And basically what you can do is that you can go beyond looking at these structures and understand really what are the biochemical structure of these communities, but you can really ask the diversity of the players within this community. So what you can do is that you can collect a sample, you can sequence, and you can reconstruct who is there and in what abundance. And when these came into play, people discovered that from this very simple, understandable structure, you have a mess. You don't basically have enough colors to represent the diversity of these communities. And so if you want, there was this new surprise of having to deal with this incredible diversity that emerged from these sequencing surveys. Now, the key point that emerged from this is the diversity of microbial communities. So this is one of the most recent version of the Tree of Life, and basically all the upper branch are different bacteria, and the one on the bottom right is eukaryotes. So all the eukaryotes you know, from us to fungi, are in the bottom right of this plot. So this gives an idea of the diversity of bacteria, and many bacteria in this diversity coexist in the same community. So this gives rise to the sort of complexity of microbial community dynamics, because we have highly complex organisms, we have complex organisms, and they change over time and they change across space because of many environmental factors. Okay, sorry, I got, what happened here? Okay, thanks. Okay. So the second challenge is that not only the environment in which they change over time, in which they these communities have these dynamics is high dimensional. There are many factors influencing them. Also, individual populations are extremely diverse, and there is a very high dimensional trait space to take into account. So really the challenge for theorists is, how do we model and we study and we build a phenomenology of communities where we have many different heterogeneous agents with many different traits, changing over time in an environment which is extremely high dimensional, where there are many factors that matters. There are many nutrients, there are many micronutrients, there are many environmental conditions, and all of these change dynamically. So what can we hope to this in time? Well, there are sort of two approaches to deal with this. One is the most traditional approach in microbial ecology, which tries to identify hidden low-dimensional structures. So perhaps we are in an environment where either over time or across space, there are some big changes of one nutrient, let's say, and these changes corresponds to one specific trait of this population, of these individuals. And so we can try to find what are the environmental gradients, the big environmental gradients, which drives a big difference in the corresponding traits, because we harness the correspondence between some big variation of an environmental axis and traits. And basically, when you try to analyze this kind of data, doing like some sort of principal coordinate analysis or principal component analysis, you exactly try to do this. You try to find the axis where the communities varies the most, with the idea of identifying a correspondence between traits and environmental gradients. Second approach is, instead of trying to reduce the dimensionality, is sort of to embrace the high dimensionality of the system, and instead of focusing on relations between environment and traits, try to focus on what are the statistical patterns of the variation we observe. And with the idea that these statistical patterns underlie some, sort of show or allow to investigate some underlying mechanisms. And this talk is about this second approach. So this is the outline of the talk. So in the first part, I will discuss some simple macroscopic patterns that emerge in natural and experimental microbial communities. And in the second part, if there is time, I will explain some new work on the statistical structure of alternative stable states in experimental communities. So as I said, for the ones that join lately, please feel free to stop me anytime to ask questions and clarifications. So, okay, so basically, the question I want to ask in this first part is the following. So in ecology and in microbial ecology, the type of data we typically have are the ones shown here on the left, where we have different species and you have different communities, which could be communities collected at different points in times. And we know what is the abundance of these different populations. Now, there are a lot of details on how these species, between quotes, are identified, but let's say that for the sake of time, we put these issues under the rug. And so we have, across communities, different species, and they vary in their abundance and in their presence. So one question that in some sense is the full, it's sort of the main question that in ecology we ask is, suppose that we have an environment, I want to know what is the composition that I should expect. So what is the probability to observe a given composition of the community if I have a given environment? And with environment, I could say I have a given environmental conditions like temperature, a given set of nutrients, a given pH, etc., and so on and so forth. Now, of course, a big challenge in this is that the environment is also affected by the composition of the community, but in general, I can imagine that I want to build this map. Now, in this talk, I'll try to do something simple, which is to forget about the environment and sort of say the environment changes. The environment changes also because the communities change. And I want to sort of marginalize over this environment. I want to ask, what is the probability that I observe a given community with a given set of species with a given set of abundances? And so this is the focus and the question I'm going to ask. So one example, if you have played with ecological theory about an expectation that we could have with this about this probability distribution is neutral theory. So if you're familiar with neutral theory, basically the idea behind neutral theory is that the probability distribution of this vector is independent of the identity of species. And so I could reshuffle this vector as I want. And the probability distribution is independent of the identity. Just for those of you who are familiar with neutral theory, this is the main, one of the main sort of result of neutral theory, or assumptions of neutral theory. If you're not familiar with that, that's not particularly important. So in this talk, I'll try to go, well, if you want, beyond this simple version and try to build a phenomenology of what is that probability distribution. So in general, this is the kind of structure that we have to deal with, and the sort of simpler question one simple question that you can ask is, if I have communities that vary over time or that vary across space, what is the distribution of the fluctuations of this abundance? So how much variation do I have? And one result is that the variation of abundance, once you rescale across species, it's sort of a very as a very reproducible shape. And the black line you see here, it's what you expect from a gamma distribution. So if you look across very different environments, across communities on the left, or you look in the human guts across hosts over time, the variation you see in the abundance of a typical species is well described by a gamma distribution. Something similar, you can observe it in experimental communities. So, for instance, in this case, you have a pool of species, and basically you assemble these communities by growing them on a serial dilution setting, where you you renoculate the next day with a small sample taken from the previous day. And at the end, you get like many replicates of these communities that were grown on from a sort of common pool on a given defined nutrient. And what you get there, it's a sort of similar pattern of abundance distribution from what you see in empirical data. So there is a common shape of the variation that we observe in the abundance over time or across space. Now, a simple way to understand this common pattern is a seemingly simple model, which is based on two very simple mechanism or processes. So one is, so this model describes the abundance of population I. So X of I is the abundance of population I. And the model takes sort of two main terms, deterministic terms, which is logistic and says that basically growth is limited, so you cannot grow forever. And there is a point where something will limit the growth of this population. And another term which is related to the fluctuations of the environment, so it's a stochastic term. This see here, it's a noise term. And the idea is that these fluctuations corresponds to fluctuations of the environment, which reflects to the fluctuations of growth rate. And that's why you have the proportionality of the noise term to the population abundance. And this simple model predicts that the stationary distribution is gamma distributed as observed in the data, and also predicts some non-static patterns in the abundance. So there is a question in the chat which says how the multiplicative noise is interpreted, and the answer is in this case I always use Ito, but if you do, for those of you who are into these things, so if you use Stratonovich, it doesn't change the prediction qualitatively. So it changes the interpretation of parameters, but you get the same stationary distribution. You can map the two interpretations into the same parameters, into the same predictions by changing the parameters, which is not always the case, but in this case it's the case. Okay, so you have this very simple model which predicts the stationary distribution, also predicts non-static properties, meaning predicts for time series what is the distribution, what are the transition probabilities, what is the probability that a given abundance, to observe an abundance tomorrow, given the abundance you observe today. Okay, so now this is about the sort of fluctuation of abundance. You can go beyond, and sort of since the fluctuations of the average abundance, of the fluctuation of abundance have this common shape, you can ask what determines the heterogeneity across species, and you can ask how the average abundance of these different species differ. Abundance around which each species fluctuate is log normally distributed across species. Then you can ask, okay, so I have this pattern of the shape of the fluctuations, the average of the fluctuation. I can ask how big are the fluctuations, and there are other statistical regularities that emerge. For instance, the variance of the relative abundance scales at the square of the mean, meaning that the coefficient of variation of these fluctuations is sort of average abundance independent. So basically, we have a very simple model which reproduce this broad, which can reproduce this broad class of statistical patterns in these ecological communities. Now, what one can do when has a very simple idealized model is to put the model to a test. And so ask, if I use this model, can I use this model as a lens, if you want? Can I use this model to identify non-trivial observation, non-trivial patterns in the dynamics of these communities, and sort of use the model to falsify it. And so one thing that we showed was that if we look at the long-term dynamics in, for instance, in the human gut, we observed rapid transitions for some species, so that there are fluctuations around a fixed value as predicted by the model, but the model fails at some specific times because there are quick transitions in the abundance. And most of the species are stable, and this plot shows the fraction of species that are stable. Most of them are stable, but there are a small minority which is unstable, and this small minority tends to shift in abundance around specific days, sort of suggesting that there are transitions in their abundance. And basically, we can recapitulate these changes by introducing sort of stochastic switches in this carrying capacity, which also explain the diversity across hosts, so how much different species differ. So this is basically what was the first part, and I ran a little bit because I wanted to go to the second one. But the main message here is that there are reproducible patterns that appear to be valid across systems, and these patterns reflect two main ecological forces. So one is that there are limiting factors, so that the growth of each populations is limited, and then that there are environmental fluctuations. That the main sources of variation in these communities, that the environment is fluctuating on a relatively fast pace, and these produce the dynamics that we observe in these communities. And then that we can use this very simple idealized model to identify non-trivial patterns and non-trivial deviations. Okay, there is a question in the chat that I'll try to open because for some reason I don't. So the question is, the noise is exploding as the X is becoming very large, or X is representing a fraction? No, the noise is becoming very large. Sorry, I need to. For some reason, I don't see my mouse. Just a second. So, okay. So, no, the noise, it's not, I mean, it's becoming very large in amplitude. So the question was if the noise is exploding when X becomes very large. Yes, the amplitude of the noise becomes bigger and bigger as X increasing, but you have also a deterministic part. So it's true that you tend to fluctuate much more when X is larger, but when X is larger, you have also a force that pushes you back on the relative abundance. So typically, what you see is that you see nice trajectory which are sort of confined. Another way to say this is that if you go to the log of abundance, the log of abundance has a dynamics which has an additive noise and a well-defined potential. So you see fluctuations which are well-defined and nothing explodes. So in this model, the dynamics of the X, which in this model represents the total abundance, it's confined and behaves well. Thanks for the question. Okay, so in this first part, I showed you that there are these consistent patterns which seem to reflect these two basic ecological forces. So if there is any question, this is a good moment to pause for me. Otherwise, I can move on to the second part. Okay. Good. Everything is clear, or everything is completely unclear? Okay, so in the second part, I want to talk to you about the statistics of alternative stable states in experimental microbial communities, and this is like very recent work that we're doing now. And it's also something where we think we have a very clear phenomenology, but we don't have a clear explanation of what we see. So we are still trying to reconcile what we see with existing theories giving rise to alternative stable states. So if you have any idea, I'm very happy to talk also about that. So this work is built on these very nice experiments performed in the group of Alvaro Sanchez, where they were basically taking inoculum, so a community from either leaves or soil, so some natural communities, and they were inoculating these communities in some defined medium, and they were growing them for 48 hours, diluting them, growing them, diluting them. And so basically this creates these dynamics. And at the end, you have a community where many of the species that you had at the beginning went extinct, but you had coexistence also of many species despite the simplicity of the model. And what was very interesting also for us was that it's very replicated community. So you have communities starting from the same pool, assembled in a very defined environment where everything is controlled, and then sort of with a fairly large number of replicates. So this was an ideal setting for us to study something similar to the statistical patterns. But also this was interesting because what it's known for these communities is that there are alternative stable states. So if you look at the final community after this time, what it appears is that, at least at the family level, so if you coarse grain these species and you look at the family composition, you have clear groups of communities which which have radically different compositions. So you have communities where there are some dominant families and others where there are other dominant families. And sort of the common wisdom about the results of this experiment is that somehow the interesting ecology occurs at the family level, meaning that at the family level you have these different alternative stable states, and within this family composition you have large variability of species composition, which is sort of uninteresting. So that basically the signal or the low dimensional ecology is at the family level, while what you see at the species level, it's very, very contingent and not very reproducible. And so in the rest of this talk, what I want to explain, what we try to do, which is trying to answer to the following question, which is, in this context where we know that there is these alternative stable states and corresponding to some coarse abundance, some coarse definition of species, what is the statistical structure of the fine-grained abundance variation? So the picture that seems to emerge is that at the family level, so at this coarse level, everything is very reproducible within these alternative stable states, while the variability within it's very wide. So what we were trying to do is to make this observation, which is qualitative, a little bit more quantitative. So the first observation that we saw looking at this data is that different species, and here ASVs, you can, if you don't know what it is, you can take it as a synonym of species, have a bimodal distribution. So each species sometimes in some communities were looked across communities, across replicates, sometimes is in high abundance and sometimes is in low abundance. And the majority of species are have this bimodal distribution. So it looks like in some communities they are in high abundance, and in some communities they are in low abundance.
Sorry, Jacopo,
Yes.
what is the difference between multiple and single inoculum?
So these are like two different set of experiments. So in the multiple inocula, basically what was done was that there were 12 different leaves, if you want, from which the community were taken. And then each inoculum was replicated 8 times. So you start from, in principle, a different pool of species.
Right, got it. Okay, thanks.
While in the second case, you have a single inoculum. So each community was started from the same set of bacteria. Okay. So the second observation is that in general, you can imagine that this bimodal distribution can be well recapitulated by the mixture of two gamma distributions. So you can sort of recapitulate the variation within each of these two bimodal parts with two gamma distributions. And sort of the picture that emerges is that there are somewhat two sources of variation in this setting, determining the variation within each of these peaks and between these two different peaks. So that somehow each species can be present at an high abundance or at a low abundance. And then there is variation within this abundance when it's at high abundance and low abundance. Now, one important point is that you could ask, well, perhaps this is just due to the fact that there is sort of the high abundant is actually saying that the species is present, and the low abundance is just saying that the species is basically on the edge of extinction, or it's very rare, and so that peak doesn't tell us anything. So actually, you can see that what is low and high abundance is very species specific. So meaning that the low abundant state for the most abundant species is an higher abundance than the high abundant state for less abundant species. So this is the first point. And the second point is that these are stable over time. So as I said before, these are tracked also over time. So you can see that species at the low abundance state stay at the low abundance state for a while. They're not declining. It's really, they're fluctuating also over time within this peak. So it's really something where the species is reproducibly present. Okay. So this is about the shape of the variation. So basically in these replicated communities, what we see is that there is these two states of abundance, and then there is variation within. So the question is, okay, so now we can imagine that the fluctuations of these species are not independent to each other because species interact, because they are subject to similar effect. They basically they respond to the environment, and they change the environment in similar or radically different ways. So what we can ask is, suppose that we have different species. Sometimes we see that their fluctuations is correlated or is independent, okay, or is anti-correlated. So sometimes when a species is more abundant, the other one is less abundant. And in principle, if we know that the variation can be disentangled in these two different states, we can argue that both the between state variation and the within state variation, so both the variation between this low abundance state and the high abundance state versus the variation within each of these two peaks can contribute to the correlation of the abundance of species. So this is to say that if you observe a distribution of correlation, so the plot on the left is obtained by looking at the distribution of correlation across all the pairs. So you have some pairs that have a very high correlation, some pairs that have a very low correlation. You can write this correlation as a contribution of being in the low versus abundance state. So let's say species 1 and species 2 can be both in the high abundance state in the same communities, or both in the low abundance state in the same communities, or in different states. And also the variation when they are both in the high abundant state, their abundance can be correlated. So in principle, both of these sources of correlation could contribute. And so what we saw is that so we can address how these two factors contribute to the overall correlation by basically randomizing the data, keeping each of these two sources of correlation fixed while varying the other. And sort of the result is that the between state variation is very correlated across communities, while the within state variation, it's not correlated. So basically we have, if we look at the data, how species vary in being in the low abundance versus high abundance state, is very correlated. It's correlated, significantly more correlated than what expected by chance, while the within state variation is not that different from what you expect by chance. So in summary, there are these two types of variation in experimental communities. One is that you have weakly correlated environmental fluctuations, well, that you have this within state variation, and sort of the interpretation for this type of variation is that these are weakly correlated environmental fluctuations, like the one I showed in the first part of the talk, with some form of limitation. And then we have this between state variation, and this between state variation is where the ecological signal really lies, where there is strongly correlated structural variation, and this corresponds to different degrees of limitations. And so the question is, somewhat the first one we understand, it's easy, simple, and somewhat boring. The second one is where really the meat, the juice is, the ecological information is. So the main result is that this between state variation is reflective of an underlying presence of alternative stable states. So if we look at these, now we sort of simplify our data structure by looking at whether a species in a given community is in the high or the low abundant state. And if we look at species, and so what we have is that we have species that are, communities that are similar in which species is present, and communities that are different in which species, we have communities that are similar in which species are in the high abundance versus low abundance state, and other communities that are different in the sort of composition, where composition here I mean whether species are in the high or the low abundant state. And basically, by looking at the similarity of communities, there is a clear block structure that emerge. So here, each row, each entry of this matrix represents how similar two communities are in these vectors of high and low abundance state. And what emerges is that there are communities which are very similar, which represents this block, and then the structure of blocks, these communities are similarly different to other groups of communities. So there is really a block structure in who is similar to whom, suggesting that these blocks corresponds to different attractors. And now this is something that emerged really at the fine level description, not at the coarse level as described before. So basically what we have is, we have these four main attractors, group of communities, if you want, that corresponds to communities that are similar in the composition, where the composition is measured in species being in the high versus low abundant state.
Sorry, Jacopo, just one thing. In the environmental data that you presented in the beginning, you don't have this sort of structure.
No.
So am I right in interpreting maybe that there are so many of these modes of alternative stable states that when you put them all together, it's just really hard to disentangle which stable state the community belongs to.
So, so in the data, in sort of environmental data, so what we see is that typically there are not that the dynamics is like around the same value. Sometimes we see switches, so that within a given host, let's say, the abundance around which species fluctuate changes radically.
But if I add more and more, let's say, gamma peaks in the middle of these two.
Yeah, but this is like something that we see very rarely.
Okay.
On the time scale we observe. So
okay.
With few, so let's say it's something that it's present. It's a small effect from the big variation that you see, right?
Okay.
So it's also there. It's just let's say as a first approximation you can neglect it. Then if you want, if you know that it's there, you can. There is a signal. It's there. Okay. So there was a question in the chat asking. Basically, the question was everything that we observe here. It's something where you have fluctuations around a steady state. Do you observe something else that is not like sort of a fixed point dynamics plus fluctuations? So I might answer to this question also at the end, but the first approximation to the answer is no. What we see is effectively, at least, and I'll explain my effectively at the end, effectively is like the deterministic dynamics pushes to a fixed point, and then there are fluctuations, there is disturbance around this. This is for the dynamics we see. We don't see limit cycles, we don't see, well, yeah, at least we don't see limit cycle. Okay, sorry for me. Okay. So the question was if there are several. There is another question. If there are several stable states for a community, could that mean that in theory there could be trimodal distribution of abundances? What is preventing this? This is a very good question. So in principle, this is what we were expecting. So if there are more than two alternative stable state, I would have expected more than two peaks, meaning that each alternative stable state corresponds to a different peak. This is not what we observe, and we don't understand why. But we try to see evidence of this, and we don't see it. So one possible explanation is perhaps two peaks are always very similar for the structure that we have. This is one possibility. But on the other hand, there could, I mean, given the abundance variation, it could emerge. So typically we don't see it. So we don't have, at least it's not something that we need to reproduce the data. So this is a very good question. Thanks. Okay, so basically we have these four attractors. And so what we can do is that we can say, okay, we can build a profile of each species in each attractor, where this profile tells us what is the probability to be in the high abundance state in a given attractor. So let's say species, a given species, if you see the color in blue, it means that it's very likely to be in the high abundance state. When in blue, it's very high to be in high abundance state in that attractor. Okay. And so basically this tells us what is the signal of each species being in each attractor. What is the profile, the barcode of an attractor. So if I want to build an attractor, when should I put a given species in the high abundance or the low abundance state? And now this, okay, it's just a set of squares colored by shades of red and blue. But then there is something interesting that emerges, that you can sort of split this profile into groups. And so basically there are different groups of species which have similar profiles. So for instance, starting from the first one, you have one species which is in the high abundance state most of the time for most of attractors, and others that are in the low abundance state for most of the attractors. So they're typically in low abundance state. Sometimes they are in high, but with no signal of the attractors. Then there is other two groups where you have species that are typically in the high abundance state, except in communities of attractor 3, and vice versa, ones that are typically in the low abundance, except in communities of the high attractor. And so you have these different patterns. So there is sort of emerging a sort of clustering of species based on their prevalence in these different attractors. And now we can go back to the first observation that I mentioned, which is that somehow there is a signal at the family level. So similar species tend, I mean, let's say there is this common wisdom that somehow the attractor has a signal at the family level, so similar species should behave somewhat similar. So now with these groups, what we can ask is, suppose that I have two species, and these two species are more or less similar. Should I expect them to be in the same group or in the reciprocal group, right? Should I expect similar species to have the same preference or not? And the first result is that the way to answer this question is that basically we can randomize species by group membership and then ask, what is the probability that two species that are very similar, that are at a very small phylogenetic distance, are in the same group? What is this probability? And compare it with the randomization. And what we find is that similar species tend to be, so pairs of species that have a small phylogenetic distance tend to be in the same group, so tend to have the same response to this attractor, the same profile. And then we can repeat for reciprocal groups. And what we find is that also similar species tend also to be in reciprocal groups, so tends to appear have the opposite preference. And so the question that we have is, well, the question. So this seems to be that there are two forces shaping these attractors at the same time. So one is ecological filtering, environmental filtering, so that somehow the same attractor corresponds to similar environments, and therefore you tend to have species that are similar in the same attractors because they have the same response, but also you have limiting similarity. So you cannot put together too many species that are similar in the same attractors because they exclude each other. And so both of the forces are appearing at the same time, are shaping the community variation, and they sort of mask each other if we don't really look at this fine-grained variation. So summary of this second part, and then I'll conclude. So there are these two, in these high replicated experimental communities, there are these high and low abundance state, and these sort of high and low abundance state have a signal of the underlying presence of alternative attractors. And there are groups with consistent attractor preference, which are closely related, where sort of species that are closely related appear both in these same and reciprocal groups, sort of suggesting that that both limiting similarity and environmental filtering shape the composition of these communities at the same time. So with that, I concluded. So I'd like to thank all the people that contributed to these works, people in my group, and other collaborators. So I'm happy to take questions, more questions now. If you're still alive.
Cool. Does anyone have any questions? They can put it on the chat.
Hi, Jacopo. Can you hear me?
Hello.
Hi. Thanks for the talk. I wonder, like, I guess here you look at in the community composition, in terms of like the species based on the DNA or something, and you see a lot of variation. But I wonder, like, what happens, like, if instead of, looking at the species, considering that, like, there's like a lot of redundancy of like, similar species can be very metabolically different, or like the same function is found across different phylogenetic. Like if this variation, what happens to these variations if like instead of looking at the composition in terms of phylogeny, you look at the like functional content of the genes, for example?
That's a very good question. So I don't have a definite answer for this. It's not obvious what to look at. So there are two parts of the answer. So one thing is that you could look at much more fine-grained populations. And this is something we are doing now. So like really trying to infer the dynamics of individual strains. This is one part of what you could do differently. And in that case, you still look at populations, and you can ask how this very fine-grained population change over time. This is one way to answer. The other one is to look at functions instead of taxonomy. The issue there is how you define functions, right? So you could say, well, you look at the compositions in terms of genes, but the composition in terms of genes tells you what organisms are able to do, not actually what they are doing, right?
Yeah, that's true.
So it's not obvious how to. I mean, it's something that let's say it's a very good question, and I would like to have a good answer. But I agree. I mean, it's a problem in the sense that we are stuck with the sort of the definition we used, and what we observed is determined by the definition we used, right? If we change the sort of definition of populations, so we could observe, in principle, very different stuff.
Thanks.
Thank you. There are more questions in the chat.
Hi.
Yes.
Hi. Can you hear me?
Yes.
So I've got a question about, it's a question about all these kind of studies where you're interested in stable states and you're using serial transfers. So how is it that you actually assess that the system has reached a stable state statistically? That's
a good point. Yes, it's a good question. So there are two things you can do. So one thing is that so there are two parts of the problem. So one part of the problem is that there is one time scale, let's say. So let me say what happens in theory and then what you do in practice. So in theory, you expect that there are two time scales. One time scale is a time scale at which the sort of total biomass of the community goes to steady state. And this happens very quickly. So this you see it in 2-3 transfers. And there are theoretical reasons why you expect this to be quick. Then what you can ask is, okay, so this is like the sort of total biomass, how big, the total number of individual total biomass. Then you can ask, how much time does it take for the fraction, the relative abundance, to go to steady state? And then this depends, in theory, depends a lot on what are the differences between populations. So if you have very small differences between populations, then this time can become very long. This is a concern. What you see in data is that often, at least this was done in the data that I presented, you track the community composition over time. And so what you can ask is, I look at the relative abundance over time of individual species, and I ask if I see a trend. If I don't see a trend in one or another direction, I see fluctuations, I call it a steady state. Which of course could also be not a steady state, could be like a transient which is extremely slow.
Yeah, I guess that was my the thing I was a bit curious about, because so you do that for all of the species, including the ones that are in that low abundance class? Because I was wondering if those low abundance ones are just sort of going extinct as a
No.
class. That they're
No, this is
definitely
something
stably
that,
associated.
this is a very good question, and they are not going extinct.
Okay.
So
Thank
you don't see, let's say, that the abundance of the low abundance state is decreasing over time. It fluctuates, you have fluctuations, but not a trend. Thanks.
Okay, thanks.
So there are a few
I-
questions. in the chat.
Oh, yeah, thanks.
were-
Oh, very nice talk, by the way. I was wondering, I listened a couple of days ago to a talk on the population dynamic by Silvia Del Monte, which was doing a similar work on microscopic heterogeneity and statistical regularities. And she called this what you can call steady state, like chaotic attractor dynamics,
Yes.
due to multiple attractors and competitive exclusion and stuff like that. And the results are basically the same because even the structure matrix is a block matrix and it's everything divided in that. So I was wondering, because in that model, there wasn't a specific stochastic term.
Yes.
I was wondering, like, how this do you think is going to change from a logistic model that gets to a chaos of multiple attractors where you have this burst dynamics, and then due to competition, they just go dormant for a while, to your model when you have, I guess, less species involved?
So this is a very good question. And when I posed during the talk answering a question about limit cycle, I was wondering whether to bring it up. So it's a good moment to bring it up. So again, there are multiple ways to answer this question. So one way to answer this question is that in some way you can interpret the model I discussed as an effective model, which describes a underlying interacting model where the sort of high-dimensional nature of interaction manifests itself in a chaotic attractor, and you sort of describe effectively a chaotic attractors as noise. Okay, so this is one way to put it. That somehow the simple picture that we see comes from this underlying chaotic attractor. Now, the problem with that is that the structure of the fluctuations you see in the chaotic attractor is, let's say, consistent. It's like corresponds to very big changes of abundance over time. So what you see is not the kind of fluctuations around the same value that we expect, but you see small fluctuations around the value and then big changes. This is at least what you get in like Lotka-Volterra dynamics, if you're familiar. So that kind of setting could explain something about the alternative stable states. And this is the sort of theory that we are trying to build, but it doesn't explain these long fluctuating trajectory around a reproducible value. Okay. So I think part of the answer could be there, but it's not the full answer in my opinion.
Thank you
But
very much.
on a very general and waving way to put it. So when we say fluctuations of the environment could be produced not just by extrinsic factors, but by the abundance, by the fluctuations of the community itself. So it could be somewhat endogenous community fluctuations. And that is the mechanism, if you want, that the chaotic attractors described by Silvia De Monte underlies.
Okay, thanks.
Thank
Now
you.
it's clear.
Thank you. Yeah, so there's some questions in the chat that were put up a while ago.
So why would the environment of different attractors be different? If I understand correctly, the experimental condition for replicas are identical. This is a very good question. So the point is the experimental condition, so let's say the environment in the sense of the nutrients that that was used in the experiment is identical. But the point is that bacteria modify the environment. And so let's say you start with some nutrients, but then as they grow, they produce other metabolites which can be consumed by other species. And now you can imagine to look at a given point in time to this community and measure the environment, whatever that means. You can measure all the nutrients that are present in the environment, pH, and so on and so forth. This environment is not just what you put it as an experiment. In the experiment is also what is produced by community members. And so the point is, the idea is that in different attractors, you have different produced environment, if you want. So that's why I was talking about different environments. It's beautiful and surprising that you get different environments starting from the same initial condition. That's very interesting, yes. So the other question was, I feel that the bimodal distribution of abundances over time could be vastly impact differential abundance analysis, as sampling could occur at different time points for the same species. Does in the low or high distribution? Do you have a comment on that? Yes, that, I mean, this is, as I said, this is quite reproducible over time. So this bimodal, sorry, perhaps I was not clear, that the bimodal distribution is obtained across communities. So if you look at different replicates, you observe different abundances in different replicates. But if you track over time a given community, so each across sampling, it's well reproducible. You don't see often, sometimes you see, but it's rare that you see transitions. I hope this clarifies.
Jacopo, I have a question.
Yes.
If you look at species level abundance fluctuation correlation, like your other paper, do you also see signs of environmental filtering?
No, in this case,
Okay.
no. So in this case, the question is, if you look at the similarity of abundance across species, you don't see it. You don't see an effect of phylogeny in this data. You see it if you really go into this detail of the abundance, but you see both the effects. So it's not something that if you look at correlation, you see a signal.
So if you take species from this reciprocal versus the same group and you look at the phylogeny abundance fluctuation pattern, then are you saying that sometimes it will be positive and sometimes it's going to be negative, such that overall it's nothing?
So yes, so if you put everything together and you ask, if I take two similar species, are they positively correlated or negative correlated? Yes, they are.
Okay.
Sometimes, exactly. So the fact that they belong to this same versus reciprocal, plus that this same versus reciprocal, there is also noise around it, cancel the signal. Yes.
Okay, thank you.
Okay. Good.
Yeah, just one last question on the noise. The noise you're using is basically like a white noise.
Yes.
Or is it direction? Okay.
So
yeah, okay.
Most of the things I showed is white. Then you can add a correlation, a time correlation. You can make it colored, and if the time I mean, for instance, the stationary distribution, if the time correlation is not much longer than the typical time scale of population abundance, so is of the same order or below, then there is a small difference, but it's not significant for the noise you have in the data.
And for the matter of magnitude is about the same as the growth rate that you have.
So if you put it the noise correlation to be the same as the growth rate, you see a difference in the stationary distribution, but it's small.
Yeah, that's why, like, to get out of equilibrium, I was wondering how much of the size of the noise you need to get these dynamics, because I guess afterwards one can include a trophic model and stuff like that, but then it's getting overlaid.
Yes, I mean, this is also related to the question. Yeah, this is exactly related to the question you asked about the chaotic attractor also. Yeah.
Yeah, I wanted to know basically how what was the size of the model of the noise that you used in the simulation to get like to similar data to compare it, because with stochasticity, you need a lot of simulation, I guess, right?
No, not Well, not really a lot of simulations. So yeah, it's not particularly hard. But yeah, the main point is that the difference with those models giving rise to chaotic attractors is that the correlation is really long range. So it's not about the time scale. It's really that the shape of this correlation, it's very broad, effectively.
Yeah, okay. Thank you very much, Kim, for the
Thank you.
talk.
Great. So thank you so much for the talk, Jacopo. That was
Thank you
really,
very much for the invitation, for the question, and
thank you. And thanks everyone for coming. So yeah,
thank you. Bye bye.
thank you. Bye.